right, I think we can get started. It's my pleasure to introduce Dr. Landweber from Princeton University today as a part of the Molecular Evolution Frontier series that's going on in chemistry and biology. And it's a nice coincidence that it's with celebrating Charles Darwin's bicentennial celebration in the case. Dr. Landweber comes to us from Princeton after graduating with a bachelor's degree from Princeton in '89, moving to Harvard University, finishing a PhD in June 1993. And she has taken up an assistant professorship at Princeton and moved up the rank to associate professorship, where she's currently at Princeton. She's going to be talking to us about epigenetics and how the interesting organism actually scrambles and unscrambles genomic information, which includes a big role for RNA. And with that, I'll leave the floor to Dr. Landweber. And
thank you.
As part of our Frontier series, we have an honorarium to present to the speaker. And this is for your...
Thank you.
Thanks for coming to Case.
Thank you. It's very nice to have the opportunity to give you this lecture in the Frontier series and to be celebrating Darwin, as you'll see, and even some of his predecessors like Lamarck might also peek into this talk. So I'm going to try to give this talk for, I realize there's people here of a variety of different backgrounds, biology and chemistry, and introduce terms as we go. And I'm going to begin by saying that I'm going to talk about epigenetics in a different context than some of you might be familiar with, but I'll also define what we mean by that as well. And there's a lot of concepts in this lecture that I'll discuss. RNA-mediated epigenetic programming and reprogramming of DNA rearrangements. So I'll talk about DNA rearrangements, how RNA influences and instructs them, and how they are epigenetically transmitted from parent to offspring, where epigenetics is telling us something about the transmission of information beyond this conventional paradigm that we're accustomed to of DNA, RNA, and protein. So we're used to thinking of the inheritance of information in a genome, this linear inheritance, as the be-all to end-all that provides the information from parents to offspring. But it goes beyond that, as many people now are learning making great strides in understanding. The field of epigenetics helps us understand, for instance, how RNA can transfer information, for instance, across generations, which has to do with gene expression. Without modifying the DNA genome, whether or not genes are on and off and when they're expressed, for instance, can also be influenced at a level that even doesn't modify the gene sequences themselves. We've known about, of course, genetic switches for a long time, but the fact that RNA, which we used to be thought of as this role here, the messenger RNA and then the intermediate and protein translation now has emerged with myriad new roles, including the regulation of genetic information. And epigenetics goes really beyond thinking about RNA, which is my viewpoint about epigenetics, my favorite way of looking at it. Many people for years have been studying epigenetics in the context of modifications to DNA. And so that raises this possibility that you are what you eat, and even your children are what you eat during gestation. And so here shown, for instance, are pomegranate seeds, which have an influence on modifications to DNA directly in yourself and also in your offspring. So people have been focusing on, for instance, how epigenetic modifications to the genome influence a lot of the variation that exists in natural populations, like variations in twins, illustrated here, for instance. So, well, in that case, there was not a lot of variation at that stage. So the idea of genes as the beads of DNA on a string providing all of the genetic information that's passed on across generations is really now considered a very traditional one. And I like this essay. I refer students to it, What is a Gene? It raises a lot of tantalizing questions. It was actually written initially in response to a discovery from Pruitt's lab about the hothead gene, which is still controversial, suggesting that there might be RNA inheritance as well. And so that RNA molecules can even potentially pass information across generations, even though normally this is thought of as the role of DNA, with the exception of things like RNA viruses. So I'll talk about that in the context of our protagonists, these single-celled protozoans. There's also the discovery from numerous occasions, including the ENCODE project that's examined in detail regions of human DNA, that information that encodes exons in the same gene could sometimes be derived from sequence information hundreds of thousands of bases away. And the last point here, sort of foreshadowed by this essay that I'll talk about, is the notion that genetic information, if it could be encoded in RNA, could be transmitted not only from parent to offspring, but even from grandparent to grand offspring through RNA molecules that keep permeating through these generations through possible pathways. And so new research into the role of RNA as a carrier of genetic information across genomes is something that's really just begun in the past few years. So first I want to introduce you to the concept of scrambled genes. And this is, again, a sort of nonlinear view of the genome, or of genomes in general. So when I first met Jonathan Gott, for instance, who's here in the audience, I was studying trypanosomes. And trypanosomes were an organism that I studied for my PhD. I love them because they have an unusual way of putting together their genomes involving massive amounts of RNA editing. That's in their mitochondria. But even if you examine the free-living relatives of these trypanosomes, for example, these organisms called dipolynemids, you'll see that there are other examples of quirky genetic architectures in their mitochondrial genomes. So quirky, so reduced in the sense of extreme reductionism, that there are examples of genes where they're encoded as separate gene pieces on different chromosomes. So that's analogous to saying a gene can be split into many different pieces, and each piece can derive from a different chromosome. Mary? Oh, it's supposed to be on. I toggled it on. Being recorded. Maybe this is out. He says it's on now? Is that better? Yeah, the switch was on. There's no light here indicating that it's on. Can you folks hear me? Because it might be, okay, I was thinking it might be the case that the recorder was picking up, but maybe not the amp. So that's all I can do. Okay. So in this case, it's analogous to, probably most of you are familiar with the notion of genes being split up into exons and introns. And so this is the sort of through the looking glass example where you can imagine each exon being present on a different chromosome. That's not the way conventional genetics certainly has emerged in most living systems. But here you have a pretty straightforward mitochondrial gene encoding the cytochrome oxidase subunit 1 that requires transplicing, or putting together all these pieces that are each derived from 9 different chromosomes just to form one gene. And it does this for each of its mitochondrial genes as far as we can tell so far. So, this is an elaborate system and not your textbook example of genetics. Otherwise, I think we'd have fewer genetics majors, right, if everything were so complex at its onset. Then there are other examples of split genes or fragmented genes. And the RNA world illustrates these beautifully. So by the RNA world, I mean the world of active RNAs. Here's just one example. This happens to be a secondary structure of the small subunit mitochondrial ribosomal RNA in the algae over here, Chlamydomonas. So it's eukaryote, it has mitochondria. But it's not just one molecule. We usually think of the small subunit ribosomal RNA and the large subunit ribosomal RNA each as distinct molecules, but this one's actually made up as 4 different molecules that can interact together by virtue of that lovely Velcro that can hold RNA-based pairs together. So this region here pairs with this one, but the same blue molecule is also docking into place, the one highlighted in orange. So this would be an example of a scrambled or a fragmented gene which has been split into 4 pieces, but they don't have to be joined together at the primary sequence level as either DNA or RNA, because they can interact in trans. So they can interact as subunits that together form the small subunit ribosomal RNA. And not to let you think that this type of system is relegated only to protozoan organisms, single-celled eukaryotes. Even if you examine some metazoa, there was a recently published case in mosquitoes where a particular gene, cross species, so it's a bona fide example, is found on 2 loci that are split onto the left and the right arm, L and R, of chromosome 2 in these various species of mosquitoes. So exon 3, in this case, needs to be retrieved from a very distant locus at the other end of the same chromosome. And so this suggests that this phenomenon is quite widespread, that actually genes can be split and then dispersed, not just split and maintained in close proximity to each other. And we call those scrambled genes. So the model organism I'm going to be talking about today is this one here called Oxytricha, and sometimes there are relatives of it that'll sneak in as well. And they fall into a group of organisms called the ciliates, which include some well-studied examples like Paramecium and Tetrahymena. Complete genome sequences, in fact, are available now for Tetrahymena and Paramecium, and drafts exist. One that we're involved with for Oxytricha, a draft I've heard of exists even for Euplodes. And they're relatives, not close living relatives, although this slide might suggest that, but they are distant relatives of the parasitic apicomplexans, like the agent, for instance, of malaria, Plasmodium. And you can see that this group as a whole, together they're called the alveolates, are sister taxa to the plants, animals, and fungi, shown here, that most eukaryotic biology studies. So here's a photograph of Oxytricha, scanning electron micrograph. And just to give you a brief introduction into its unusual genetic system and its elementary architecture, well, to start off with, it's not just a conventional eukaryote and having a single nucleus. It has two nuclei, or sometimes even more than one nucleus, or more than two nuclei, but two types. There's always some micronuclei and also some macronuclei. Sometimes they have two of each, sometimes they have 100 of one, and a small number of the other. So there's a lot of variation that's possible in these organisms. And what you can see here is that they differ in size both physically, that's because of DNA amplification in the macronucleus, and then they differ in sequence complexity because of genome reduction also in the macronucleus. So the macronucleus is a highly derived and highly processed version of their germline, which is the micronucleus. So gametes, for instance, are formed from the micronucleus, but day-to-day transcription comes from the somatic macronucleus, which is really like an organelle whose sole function is to form messenger RNAs and functional RNAs, and basically to allow the daily performance of the cell to take place. So some other features about this genome architecture, or this minimal genome architecture that's formed in the macronucleus, is that it extracts the coding information that's highly dispersed in the germline genome, okay, which are, so you basically have short islands of coding information and long oceans of non-coding information. The coding segments are referred to as MDSs for macronuclear destined sequence. They're analogous to, and were initially called in the literature, exons, but because they're spliced as DNA as opposed to being spliced as introns, it's not appropriate to refer to them as exons. I'll try to just call them the coding regions most of the time, although they can also themselves carry an intron, so they're not all coding, or they can also carry regulatory regions. And then the intervening sequences that separate these, analogous to introns, are called the IESs for internally eliminated segments. And there's also spacer DNA and intergenic DNA. And in total, the sum of all of the IESs plus the intergenic spacer DNA is about 95% of the genome. And that's actually very similar to the human genome, which is represented by approximately 1 to 2% exonic DNA. And the rest is also called non-coding, possibly junk DNA, although approximately 50% of the human genome seems to be derived from transposons. And we're learning more about the functions of much of the rest of that non-coding DNA as well. So in the unusual case of Oxytricha, they have this alternative lifestyle where they have their own solution they've come up with to this paradox of why so much junk DNA in our genomes. We can't answer the question yet as to why they have so much junk DNA, although we'll come back to that at the end. But we can answer the question, what do they do with all this junk DNA? That is, on a day-to-day basis for vegetative growth, they throw it away. They are, the premier recyclers. They toss out 95% of their genome, everything that's shown in light blue, and package only the remaining dark coding segments together to form the functional genes. And then they package each with a short 20 base pair telomere. So a lot of telomere biology was revealed first by studies in Oxytricha as well, because they are paragons for telomere biology, because they put a telomere at the end of just about every gene, not just every chromosome, but they tend to have this one gene, one chromosome paradigm. And sometimes there are exceptions, 2, 3, 4, 5 gene chromosomes, but most genomes, or most of the genes, are found on simple 2 kb miniature chromosomes that we call nanochromosomes, because you can't get much smaller than one gene as a coding unit. Okay, you can get smaller if you go to diplonemid, which has, one exon equivalent per chromosome. But in this case, you have basically very simple units. Sometimes a nanochromosome might encode only one tRNA, or sometimes they might encode multiple genes. So there's this extreme genome reductionism that takes place in the formation of the macronuclear genome. And also, correspondingly, there's a massive number of chromosomes on the order of over 20,000 chromosome types in the macronucleus because they're typically one gene per chromosome. Now, because coding segments are dispersed over the original germline nucleus, they have to be fused together, and sometimes they can be fused together across loci or potentially across chromosomes as well, like in diplonemid. And what you have taking place is the opportunity to permute or shuffle the order of these segments as well. So this is indeed an illustration of what we mean by scrambled genes. We have segments 1, 3, 5, 7 upstream of 6, 2, 4. This is just an illustrative case, but basically there's the opportunity to shuffle and mix and match the coding segments derived from the germline to form the possible sets of chromosomes in the macronucleus. So during the life cycle of a mating pair of ciliates, so here's your X-rated slide in the upper left corner, a conjugating pair of oxytricha. And during their life cycle, mating or conjugation triggers meiosis of the gametes. Okay, so meiosis of the diploid micronucleus to form haploid gametes. Then there's destruction of all but one, and they then differentiate into a stationary micronucleus and a migratory micronucleus, at this point each haploid, which migrate across the bridge between the two conjugating cells. So what's left are two genetically identical X conjugates at this point that have a newly formed diploid zygotic micronucleus. So this undergoes one more cell division, or one more nuclear division over here, to form two equal micronuclei at this stage, one of which differentiates on to become the new macronucleus. And at some point in this stage, actually a little bit later on, there's the destruction of the parent nucleus. Okay. So during this process of conjugation, there's a stage right here where there's massive genome differentiation. And during this genome differentiation, there has to be the opportunity to completely rewrite the genome sequence information from the micronucleus and transform it into that miniaturized and then subsequently amplified one gene, one chromosome genome in the macronucleus. So I can also give you a snapshot about what we know about the genome sequences so far, since we have genome projects underway for both the macronucleus and one about to begin for the micronucleus. So in the macronucleus, as I was saying, we have this massive number of chromosomes, over 20,000 chromosomes, each of which is present actually at a high copy number because of gene amplification, high copy number, about 1,000 copies each. And this is why the macronucleus itself is larger, even though it has smaller sequence complexity because of that high amplification. And comparatively in the micronucleus, we have a lot of repetitive DNA. What is shown here is that there's approximately 5% so far of known transposons replete throughout the DNA sequences. And what fascinates me, what draws me to this problem, is the fact that approximately 1/4 to 1/3 of all of the genes in the genome require descrambling or reshuffling of the order of those coding segments. So we have basically the unusual opportunity here to have a comparative genome project within a single cell, right? Because it's one cell, two genomes, and we're really fundamentally interested in the mapping of each genome onto the other and the genetic and developmental processing of how the second genome, the macronucleus, forms from the micronucleus, how that's regulated, and how it's evolved over evolutionary time. So to give you a snapshot of some of the actual illustrations of different scrambled genes, because I started off with a sort of pedagogical case, in the example that I think was first discovered in David Prescott's lab, I think the first one he described was the actin-1 gene, where he coined the term first exons and introns for these different DNA segments, and then later changed that to MDSs and IESs. David Prescott at the University of Colorado at Boulder originally discovered these phenomena and characterized these scrambled genes. And so in the case of the telomere end-binding protein alpha subunit, or actually the alpha telomere end-binding protein, what you have here is an unusual strand asymmetry where there's a collection of odd coding segments, or MDSs, located upstream of the even ones. So you have 1, 3, 5, 7, 9, 11, 13, followed by 2, 4, 6, 8, 10, 12, 14, and then these over here don't require any unscrambling. So we would call these non-scrambled MDSs or segments at the end. And so this affords the unique mode of descrambling, which can exploit the structure that's available to the DNA sequences. So you can have, for instance, 5, once it's in position between 4 and 6, brings into proximity 2 between 1 to 3, and for instance, 11 between 10 and 12. So there's an opportunity for shortcuts to take place in the ability to descramble these gene sequences. And we don't know why it evolved this unique segment orientation of having odd segments clustered from even segments, but we can visualize mechanisms that will propagate that pattern. Then a simpler case is the actin-1 gene, which has actually a cluster of non-scrambled segments or non-scrambled MDSs, 3, 4, 5, 6, 7, and 8, all adjacent to each other. M just stands for MDS here. But then segment 8 is followed by 10, which is followed by -2 and -1, so that means they're on the inverse complementary strand, they're reversed, followed by segment 9. So actually descrambling of this gene is relatively simple in that you only need to have an event that transposes segment 9 and also inverts and translocates segments 1 and 2 together. And then the last example I'll illustrate here is another well-studied case of DNA polymerase alpha, also initially discovered in David Prescott's lab. And in this example, many of the cases of this gene and its orthologs, which are its relatives in other species, are scrambled in 48 to 50 pieces. And so this is getting indeed more and more fragmented, and sometimes these fragments, like the ones on the red locus, are found on different loci, which have a signature of genetic crossing over between the two. That is, crossing over happens in the micronucleus between them because they are on what we can define as genetically distinct loci. And here, everything that's highlighted in blue is on the opposite strand with respect to everything highlighted in green. So again, you have strand asymmetry, whereas opposed to, or usually we have a conventional strand asymmetry would be all the exons are on like the top strand of a strand of DNA. Let's say they're all on Watson. And here, you lose that strand asymmetry because you have some segments encoded on the Watson strand and some segments encoded in the inverse complementary Crick strand. And so that is something which is not usually found in biological systems. So that's not the most elaborate case so far. Just to give you one example that goes beyond that, an undergraduate in my lab, Genevieve McQuillan, who's now in medical school, was originally studying for her senior thesis the ribosomal RNA locus, one of the best studied loci across all life on Earth. It's commonly used as a marker, for instance, for DNA barcoding, because every known example of life on Earth, apart from viruses, carries a ribosomal RNA gene. And this ribosomal RNA gene she found was initially scrambled into 150 pieces at our last count. And so these pieces can be shown, she likes jigsaw puzzles, her father told me at graduation. So she took this jigsaw puzzle and first could characterize approximately the first 50 pieces, for instance, of the ribosomal RNA precursor gene in the germline micronucleus. And at present count, we have basically 4 main loci that each contribute approximately 30 coding segments each. One of them is a little bit smaller. And those that are shown in italicized font are the ones that are on the opposite strand. And those that have an asterisk next to them are actually ones that have paralogous or duplicate copies elsewhere in the genome. So, and these are 4 orphaned sets of loci at the bottom. So we have basically these 4 major loci, each encoding quite a number of coding segments, or MDSs, and then 4 smaller contigs that together comprise approximately 90% of the gene, leaving about 10% of it still unmapped, which will probably defer actually mapping until we have a draft sequence of the micronuclear genome, because some of these pieces are just very small and you're not going to be able to recover them, for instance, by PCR. So Oxytricha's genome is a puzzle. And when we examine this puzzle, some of you might be familiar with Rube Goldberg cartoons. Basically, its genome is like having these pieces that have to interact with each other to form one functional unit, in this case, one functional gene that can encode a protein. And we want to try to figure out what are the rules that put all these pieces together. So one of the sets of rules that we've been studying are simple word sequence repeats, direct repeats found, for instance, at the end of each segment that provide a go-to command to go to the next copy of that direct repeat in another segment. But I'll show you that this level of information is far from complete. Although in the context of Rube Goldberg types of cartoons, you have the ability to imagine how each step links together the next step in the process. Well, the cascade of genome rearrangement is something that we've been studying so far at a microscopic level of single gene rearrangements. We want to understand, like I was saying, what are the rules that allow all these pieces to work together in a smooth fashion. And that is, what is the scaffold that provides all the information to link them together? So one thing I'll show you is that some of this scaffold and information is provided by non-coding RNA molecules, a new class of long non-coding RNAs that we call template RNAs. And subsequently, they provide the information of where to link everything together. And we know some of this by having done experiments where we mutate the RNA and provide a different alternative template that I'll show you. And that tips the balance in the favor of alternative rearrangement pathways. So some of the information I was saying comes from word sequence repeats. And here are examples of pointer sequences found at the end of every segment. Here's an example at the end of segment 12, found at the beginning of segment 13. Most of these sequences are relatively unique, meaning they exist as 2 copies locally. We don't know how many copies they might be in the whole genome. But some of them are redundant even in the context very locally, which means, for instance, this pointer sequence found in 12 and also at the beginning of segment 13 is directly contained, embedded in the sequence repeat or the pointer that says go from 31 to 32. So that raises the possibility of having alternative rearrangements take place that leap from 12 all the way to 32. And I'll show you illustrations where that happens as well. We don't know about this particular example, but we know that often these pointer sequences are far from complete in providing the genetic information for restoring a functional gene, and that cryptic pointers, cryptic repeats throughout a gene, can serve as landing docking sites for recombination to take place between them as well. So to understand the processing of these genes, one approach is to examine developmental time points throughout the whole process of rearrangement. And so we chose to do this with that simplest example of a scrambled gene. We're not going to tackle this kind of question with the 150 segment scrambled gene, but this one for the actin-1 gene required only a small number of descrambling events, right? Because most of these MDSs at the 5' end are non-scrambled. So descrambling the actin-1 gene, we found, can proceed through a series of steps as shown here. So this is our Rube Goldberg cartoon expanded. Okay, this is a view of what's actually taking place. So early in development, what we see happening first is mostly removal of the conventional IESs, or removal of the interspersed DNA between coding segments. So we have here, for instance, joining segments 3, 4, 5, 6, 7, and 8 happening nearly all at once. It's not always the case that the DNA segment, for instance, between 3 and 4 is removed, before that between 7 and 8. Some of that seems to be a random choice. But we see pretty soon in development the emergence of a simplified version of the precursor gene that's lost almost all of its linear non-coding DNA strands. So lost all of its non-scrambled IESs. And then what we see happening, and we find evidence for all these steps by PCR, is translocation of segment 9 can occur between 8 and 10. And you can find evidence for that in the context of intermediates or potential intermediates or partially rearranged, partially edited molecules that have segment 9 found in between 8 and 10, but they still have 1 and 2 inverted downstream of segment 10. And on the flip side, there's evidence for the alternative pathway taking place, which first takes segment 9, 1, and 2 together. I like this one, and transposes it to the 5' end of the gene as a unit. And that produces putative intermediates, or partially rearranged molecules, that have 9, 1, 2 found at the 5' end of the gene, although we don't know if segment 9 might also be present in between 8 and 10, since this part of the molecule couldn't be surveyed at the same time. And then subsequently, if this step is chosen first, then the next step in this pathway is to translocate segment 9 to where it belongs, and likewise over here, the converse step can happen next to form a functional macronuclear product. Now, the additional point added up here is that there's a lot of error that takes place at this first step, and the error is always of the type where incorrect pointer sequences are chosen. We call them cryptic pointers, that will fuse basically anything to anything where there's been a cryptic pointer present. And I have some examples on another slide after this one, but this just serves to underscore the fact that a simple pointer sequence that's often in the length range between 2 and 14 base pairs is rarely sufficient information to always accurately derive, for instance, the fusion of segment 4 to segment 3 in this case. So here we can look at a snapshot of some of those errors. And when you examine the errors, what you should see here is the removal of the sequence in red. And here's the correct pointer sequence, annotated P, of the 5 base pair repeat TCGTT at the 5' end and at the 3' end of the IES. Also at the, you can see it's at the 3' end of the upstream MDS and the 5' end of the downstream MDS. I'm sorry, I should have said MDS up there. M annotates the NDSs, I annotates the ISs, and P for pointer. And then what's shown in white are opportunities for cryptic pointers that have been used by the system instead. Early in development, we see basically a whole range of sequences deleted. It's consistent with the idea that comes from tetrahymenas experiments, that there's probably some epigenetic marker on the DNA sequences to be deleted. And here by epigenetic, we mean modification of the DNA, in this case, modification of the histones that recognize that DNA. In fact, in tetrahymena, if you modify histone H3 at lysine position 9 with the methyl group, then sequences associated with it become deleted. And so if there is some specific epigenetic mark on DNA sequences shown in red to eliminate them, like a copy editor, delete those sequences, then the next step may be scanning for sequence repeats flanking such a region, and then it needs another step to add fidelity to such an information system. Otherwise, it can often, as you see here, make the wrong selection. So that invoked the idea that there needs to be some kind of proofreading or error correcting mechanism tacked onto the system. So the key way we've been thinking about this, we and our colleagues like David Prescott and 2 computer scientists that he collaborated with, was that templates might be providing the missing information. And by a template, we mean something like either an old relic DNA copy from the degraded parental macronucleus, or an RNA copy that could be providing this information as well. And so if templates are simply derived from copies of the maternal parent's genome, and if they get transported to the newly developing macronucleus, then they can lay out a scaffold on which to provide the information just to properly guide the rearrangements, for instance, in the events that require simple DNA deletion, and also the events that require fusion of dispersed DNA segments, leading to formation of the correct product. So analogously, you can think of this as, if you're tackling a jigsaw puzzle, one strategy might be to simply flip it over and look at the cover of the box so that you know what your ultimate goal is. And so that's analogous to the type of rearrangements that would be template dependent. They're examining a copy of the parental version to see, they're cheating. They're going back to simply copy somebody else's homework who got it correct. So Mariusz Nowacki, an extremely talented postdoc in my laboratory who spearheaded this effort, started these types of experiments by seeing if we could disrupt the putative template RNAs. So this is an example where we tried to use RNAi against non-coding RNAs, something that some people have found difficult for small non-coding RNAs, but we found seems to be effective for long non-coding RNAs. And so the use of E. coli expressing to short double-stranded RNA molecules, long double-stranded RNA molecules that would subsequently become diced into shorter double-stranded RNA molecules can destroy those template RNAs. And subsequent to that, the hypothesis was that this would halt or abrogate DNA rearrangements if the template RNAs were reduced in their availability. And basically, we tried this on different genes, and that's exactly what we saw, is that the new macronucleus, as it was developing, contained errors. And some of these errors are retention of the precursor micronuclear sized DNA, for instance, where here it's actually smaller because the segments, since they're out of order, allow you to design PCR primers that produce smaller products from the precursor than from the product version, even though there's actually a lot of DNA that should be deleted from the precursor version. And so here, this is unusual in that there's retention of the micronuclear size product and very little production of the correct macronuclear size product for the DNA polymerase alpha gene. And that was when we treated with RNAi against the Pol alpha gene itself. And so correspondingly, if we treat against, use RNAi against, it's either abbreviated TBP or TEBP alpha gene, you can see here here, there was a more modest effect, and we saw, however, accumulation of lots of aberrantly sized products. So when we clone and sequence a lot of these aberrantly sized products, what we find is that there sometimes is a mix of correct rearrangements having taken place and partial unscrambling of genes, for instance, fusing together segments 6, 7, 8, 9, 10. Remember, this is not a knockout of the templates, but there's simply a reduction of template concentration in these types of experiments. At least that's our assumption. And what we saw subsequently were also some correct permutations or some correct rearrangements taking place and some gross errors taking place, like duplications of segments within a gene or deletions of any portion of a gene or any portion of the gene that has these cryptic pointers flanking it. So for example, it can leap from segment 8 all the way to 13, not exactly at their ends because the pointers are different, but fusing these things together, those cryptic repeats in the genome. So lots of errors that were not being corrected. And similarly, if we examine the DNA polymerase alpha gene, where we saw there was mostly retention of the precursor size product, when we did look at what types of processing was occurring in this gene, and remember this is one of those more complex genes that's broken into 48 segments, we found that basically there was mostly just cryptic deletions that were taking place at those alternative pointers, and sometimes gross deletions, for instance, leaving most of the gene absent. So the summary here of this first experiment was that clearly this led to our first opportunity to disrupt the whole phenomenon of gene unscrambling and then to begin to dissect it molecularly. So clearly one of the next steps that we wanted to do was see if we could find these elusive templates. We had first sort of sought them by disrupting them, as geneticists often do. We don't have classical genetics in Oxytricha, but we now have the ability to go in and bash things using RNAi. That stands for RNA interference, and is something I alluded to at the very beginning in that definition of epigenetics, the discovery that received a Nobel Prize just a few years ago, that RNA molecules can eliminate messenger RNAs, so not just turn off gene expression, but actively be involved in the destruction of RNAs that are present, or even non-coding RNAs. They don't have to be messenger RNAs. So we simply use telomere reverse transcriptase PCR, and by telomere I mean PCR from the telomere to telomere, to see if we could detect the presence of these elusive templates. And what we found is that actually right at the beginning of mating, 5 hours or T5 after the onset of conjugation, so when we see pairs forming, then you can see the presence of these templates from top and bottom strand, sense and antisense strand, for each of 3 different target genes that we surveyed. They do contain introns, so these sequences are clearly not messenger RNAs, not messenger RNA derived, and they also contain sequences all the way out to the telomere, so that includes the normally non-transcribed portions of the molecule as well. So one thing we found is if we examine what's going on just 15 hours later, there does seem to be conversion of these template RNAs to a new family of small RNAs that we haven't yet characterized, but which all appear to derive, in sequence at least, from the macronuclear genome. And so this raises the possibility that actually the templates might be active as small RNAs, and we haven't yet deciphered whether their mechanism of action takes place as small RNAs or as long template RNAs. But we know that the sequences of this class of small RNAs is generally derived from the macronuclear sequences, so that's consistent entirely with a model where there's complete transcription of the macronuclear genome into double-stranded RNAs starting at the telomere, ending at a telomere, suggesting that telomeres can act as promoters, and then those RNAs can be diced to form these small RNAs. And so another student in the lab who recently graduated, is now a graduate student in systems biology at Harvard, wanted to tackle this system with a little more chutzpah. He said, let's just see if we can make radically alternative templates. And to us, it's radical because we've switched the segment order in the middle of the gene, which is going to affect the function of the protein. It's actually going to create premature stop codons halfway through the protein. So we're just completely throwing a wrench in this system if it works, and using this to reprogram DNA rearrangements. So instead of having the normal order of coding segments here, 1 through 7, this is taking a gene which is normally not scrambled, so the TEBPB beta gene normally has no requirement for any permutations, but just conventional DNA IS deletion. And instead, we're turning it into a scrambled gene by asking it to permute or to flip the order of segments 4 and 5. And we did so by now exploiting the presence of repeat sequences in the gene to select which regions we wanted to use as all our alternative pointers. And the nice thing about this experiment as well was that we could have a convenient short assay for whether or not the rearrangement had taken place according to the wild type version or the switched or reprogrammed pathway, because by simply measuring by PCR the distance from segment 4 to the end of the gene, you would see a shorter product if segment 5 is now located upstream of segment 4. So basically what we observed was that when we construct alternative chromosomes, okay, so we first did this as a DNA microinjection experiment, where the alternative chromosome contains the reversed or switched order of segments 4 and 5, and inject this into the developing nucleus, actually to the parental nucleus of a conjugating pair of ciliates. And so this would have the opportunity to be treated like an endogenous or a native chromosome and undergo processing and influence the processing just as any other molecule might. So we're essentially changing, if this, I should go back to that Seurat painting and, transpose two people in that particular painting so that they're in different positions. And that's analogous to what we would observe here. And so what you see is that the offspring, or the F1 of this mating cross, sometimes followed exclusively the new paradigm. So we reprogrammed them by providing alternative DNA nanochromosomes in the parent's macronucleus. And sometimes we see cases where they followed the switched rearrangement pathway in what sort of appears to be like a heterozygous behavior, in the sense that they sometimes retain the wild type version and sometimes produce the switched or newly rearranged version. This type of cell we predict would have a better survival opportunity than a cell that follows the entire new pathway, because this cell couldn't be making new versions of functional TBP beta proteins. What was striking about this result, though, was that when we examined the offspring of these cells, we saw retention of the new pathway, even though the treatment was only applied to the parent cells. Okay, so this is now reaching to the F2. We also show that it still gets murkier, but it does propagate to the F3, because the reason is you see a reversion happening over here to wild type, and that's presumably because of the pejorative effect of having a dysfunctional TEBP protein. So this raises the possibility of epigenetic inheritance, so non-DNA inheritance, in this case happening from generation to generation. The genome, we've confirmed, of the F1 and the F2 has no changes in the micronucleus. But there's still this change now in the macronucleus, especially in this one, which is still following predominantly the rearranged pathway. So it's ironic that while we're celebrating, of course, the 200th anniversary of Darwin's birthday and the 150th of the publication of The Origin of the Species, it's worth remembering also that Lamarck's publication of the first formal theory of evolution, Zoologique Philosophie, actually promoted the idea of the inheritance of acquired characters. It wasn't actually his own original idea, it was the thinking at the time, but he extended that into a theory of evolution where the use of a part can transmit to offspring more frequently than a disused part would. And so this would be an example where RNA is a perfect measure of the use of a gene to a certain first degree. And so in fact, if RNA species are more abundant for a certain rearrangement pathway and get passed on to the offspring, then this can happen in a way in which it can afford an opportunity to transmit acquired information over a lifetime. So if it's more valuable to make a certain RNA for a certain pathway at one stage, that can be passed on to the next generation. So this summarizes this observation so far, that the programming can be epigenetically passed on to offspring. We don't know how epigenetically stable it might be, because this set of experiments that we did produced a crippled version of essential protein. And so in the next set of experiments that we want to do, and we're looking for people to do these types of experiments with us, we want to test whether actually epigenetic information can be stably inherited via these RNA rearrangement pathways. And by doing so, we'll redo these types of experiments with a neutral marker where no actual changes to a protein take place. So lastly, in this part of the talk, we wanted to, of course, show the sort of final experiment in this series that the templates can be active as RNA molecules, and you do so simply by microinjecting RNA versions, which can come from sense, antisense, or their combination, which has the potential to be double-stranded RNA. And this did indeed recapitulate the observation that we can have rearranged pathways, new rearrangements taking place when we provided the alternative RNA template. So this work was published now over a year ago, and so you can read about it, and there's also a YouTube video posted on my website, and I'll show you some snapshots from that later on. But another observation we made in this paper, so buried in one paragraph of this paper, was a strange occurrence we found of occasional sequence information passing from the RNA template or the DNA template to offspring as well. So this raises the possibility that another way in which information can be passed from parent to offspring, not through DNA, but through RNA, so it's again epigenetic, is that point substitutions that occur in a somatic lineage. So you tend to think of often DNA point substitutions that occur during our lifetime can contribute to late onset genetic diseases, for instance. But sometimes a somatic change that arises in an organism's lifetime could also confer a selective advantage. And oxytricha would be poised to actually capture and drive to fixation some substitutions that can occur somatically, such as this C to T substitution found here, where there's a T in the templates that we provided and a C in the wild type sequence. And the reason we put substitutions in these templates was so that we could clearly distinguish endogenous from the injected artificial molecules. But this particular point substitution actually transferred to all of the rearranged molecules. And you can see it's located just 4 base pairs from the end of segment 4, so right next to one of these homologous recombination types of junctions at the pointer. And like there's this sort of trickle down effect that you see 12 base pairs upstream. There was also another point substitution, A to T, which transferred with much reduced efficiency. And that's consistent with polymerase activity in the vicinity of these DNA rearrangements. And that's consistent with the need for DNA proofreading, where there could have been errors, for instance, in the selection of incorrect pointer sequences. And if there's a polymerase that's really active in this region, then it would have to be an RNA-dependent DNA polymerase. We have no evidence for reverse transcriptases, for instance, in the oxytricha genome so far, or bona fide ones, I should say at least. But Michael Resnick's lab had recently shown in yeast, and now I think it's found more broadly, that there can be RNA-guided DNA proofreading that seems to be mediated by DNA polymerases. So the delta and epsilon DNA polymerases seem to have an ability to also read RNA oligonucleotides. And so those would be our candidate genes for what might be contributing to the polymerase activity that might be required to repair incorrect DNA sequences at these rearrangement junctions. And this frequency where the C to T transition right near the end of segment 4 transferred to all rearranged molecules, and the nucleotide substitution A to T, 12 bases upstream, happened with decreased efficiency. That was entirely recapitulated whether we did a DNA or an RNA microinjection. So it's been affirmed. So more recently, we wanted to query what kind of proteins are involved in mediating these DNA rearrangements. So we now have a candidate. We want to go study RNAi knockdowns, for instance, on the epsilon and delta DNA polymerases. We already examined the alpha DNA polymerase because we had that construct for RNAi readily available, and it had no effect on DNA rearrangements so far. So that's one candidate gene out, but we have many more to try. So we also thought that maybe a class of genes that could be involved would be transposons. So just like in the immune system, VDJ rearrangements are catalyzed by proteins that are themselves derived from domesticated transposons. We thought that maybe some of the transposons that are replete throughout the micronuclear germline genome could be contributing to the DNA rearrangements in this organism. And so we examined the 3 studied classes of transposons and their encoded transposases that had been described earlier from Glenn Herrick's lab in oxytricha. And first of all, anybody who's an oxytricha aficionado might recall the general thinking that there was thought to be no transcription at all from the micronucleus. So the germline is thought to be relatively silent and only used for passing on information, the genetic information from parent to offspring. And what we found instead is that there is really quite timed explicit expression of the transposase, at least from these transposons. They also encode 2 other proteins at approximately 24 hours into development. So this is after the presence of templates and after their conversion to small RNAs, so putting this all into a developmental timeline. And what we found was also that there are 3 existing classes of these TBE transposons that all themselves can diverge. They diverge as an ancient family of transposons or ancient sets of transposons, at least 10 to 20% within any of these individual groups. And so this simply shows that they are all expressed at this similar time window relative to a ribosomal RNA control that's expressed constitutively. And you can also observe them. So up here you have northern analysis, and down here reverse transcriptase PCR. And then what we next observed was, we asked the question, what would happen if we used one of our favorite tools, RNAi, to reduce the concentration of the TBE transposases? And what was a little difficult in this particular experiment was that we weren't using RNAi to disrupt a single type of sequence, but we were trying to use RNAi to actually silence 5% of the genome. So that's quite different because we anticipate that there are thousands of copies each of the TBEs 1, 2, and 3. So if we sequence any randomly sampled set of TBEs 1, 2, and 3, we rarely get the same identical sequence because there's thousands of copies per the genome, so they're replete with sequence variation. And so what we did was we chose somewhat randomly 4 representative sequences from each of TBEs 1, 2, and 3. TBE, the name of the transposon family, stands for telomere bearing element because they contain the T4, G4 telomere sequences within them. And then we treated developing cells with either just the 4 TBE1 constructs or the 4 TBE2 RNAi constructs or the TBE3 RNAi constructs or any pairwise combination, so that makes 8 constructs together, or all 12 constructs, which would be a triple RNAi silencing experiment. In this case, we, rather than feeding the cells E. coli expressing the RNAi, here we were actually microinjecting them so that we could ensure stoichiometry retained for each of these combinations of different RNAi vectors. And so what we observed here was that, like in the case when we disrupted a specific RNA template and saw specific disruption in the rearrangement of that gene, now when we survey any gene, this is 4 representative ones, the same genes I've mentioned so far in this talk, any gene that we survey, we see there's been accumulation of the precursor product and basically a halting of or a stalling of the full rearrangement process, only in the case of the triple RNAi silencing. So only when we eliminate all of the classes of TBEs 1, 2, and 3, or reduce them, of course, I should say, since we can reduce their availability approximately 6-fold, but we don't know, genome-wide. We don't have the ability to knock them down completely, and if we did, the cells would probably not survive. So it's one of the beauties of RNAis. They're really giving you conditional knockdowns. And in addition, we saw a general phenomenon of incomplete DNA elimination. So in general, more incomplete DNA processing taking place when we treated with RNAi against TBEs 1, 2, and 3. And we saw some effect over here with TBE 1 and 2. And what you're seeing here is that there's a more intense staining of the micronuclear DNA. And so there's accumulation of the precursor, again, and relatively little formation of the products, which would be the MAC DNA. And in addition, as is not surprising, this is really kind of a control, we also see less elimination of the TBE transposon, as indicated here by probing the germline DNA with a probe for one of those classes of transposons. So if we look at specific cases, we saw the same recapitulation of the same types of errors happening in DNA rearrangement consistent with stalling the process. So you see examples of aberrant rearrangements happening, some of them happening multiple times, where there's been incomplete processing and there's retention of some of those IESs and correct deletion or even rearrangement of others. And likewise, in the second locus shown over here, you can see illustrations where there's just almost complete processing, but retention of some IESs in just about any surveyed sequence that we found. So this is work that was actually just published in Science Express last week, so that's why I stuck this slide in at the beginning of the talk, so that you have the opportunity to see more about this type of work. And Also, I indicated there are some YouTube videos. We actually didn't upload these ourselves, but New Jersey Network News did for us, which illustrate the microinjection directly into the cell. So these are great in terms of teaching about the system, and they show the system and the microinjection apparatus and also the people involved. I want to thank, and although it's hard to find, the actual YouTube video, you need to search. I didn't even know it was there. Like I said, it's not available by searching my name, but if you search DNA bypass, you'll find it. Or you can simply get it from my lab's website. And so I want to thank lots of people that contributed to this work. And also acknowledge, of course, Darwin in his bicentennial year. And here we have the current and former lab members that contributed to various aspects that I talked about today. And collaborators shown on the left, including the Washington University Genome Sequencing Center, who's sequencing Oxytricha's dual genomes with us. And collaborators have included the LIPS lab in Germany that did the time course analysis of DNA rearrangements with us. A computer scientist named Jegorz Rosenberg, who's very interested in modeling this type of system as computation. My lab really tries to combine an interest in bioinformatics and computational biology with the experiments that I talked about today. And David Prescott, who really founded this field, and so I thank him for that. And I thank all of you for coming today and for this invitation. Thank you. It's a lab group.
We are the ones who are strange. This is the prototype. This is how it all started. And that putting the modules together in different combinations, eventually they fused and became the whole genes that we have now.
Right, one way of thinking about this is it might be recapitulating atavistic processes like those that could have contributed to the evolutionary formation of genes in an earlier world. The catch is that while the processes might be similar and it could be analogous, a phylogenetic reconstruction of the evolutionary timeline of these DNA rearrangements that we did earlier supports the idea that this particular deconstructive genetic system is derived. So it itself is not an ancient genetic phenomenon. Is derived from a normal, more conventional genome. Still, there's a so that it's derived from an ancestral genome, which itself was a well-formed eukaryotic genome. This doesn't have anything to do with necessarily life's origins or even the origins of a eukaryotic genome. It just means that this particular genetic system itself has specialized. And so some of us think of this in terms of Muller's ratchet, that once a system for error correction is available, then more and more errors, like more and more DNA rearrangements, can crop into the genome, and each one becomes effectively neutralized by the system that permits faithful error correction across generations. So it's probably a system like RNA editing, which itself capitalizes on parts, within the machine, within the genome parts list, that could have evolved themselves initially to help construct the integrity of linear genomes, but are now able to work with these deconstructed genetic systems. In the back.
As I understand, the point of your lecture was that the genetic information is carried somewhere else besides the chromosomes and the ribosomes of the RNA.
Or it can be. It's a different role for RNA besides its conventional role as mRNA and tRNA and many other functional RNAs.
So you think that RNA can also carry genetic information?
Yeah, so a good example was when there was a single base substitution, where if you introduce an RNA with a T at the same nucleotide where the genome has a C, then across generations you can see that C to T change has occurred. So even at the base pair level, there can be a nucleotide substitution where the vehicle for that change is an RNA. The DNA sequence in the parent's genome and in the germline genome is still the C, but what's expressed and produced into mRNA is the one containing the substituted nucleotide.
Could this also occur in humans?
We're examining whether the general phenomenon of RNA-templated DNA rearrangement might occur in human cells, but we don't have results we're confident in yet.
And then I have a third question, if I may. Go ahead. is the idea to sort of scatter the risk of damaging the real important part of DNA that's carrying the information. So to spread the information on DNA into a larger area so that the risk would be reduced, the risk of damaging.
Right, well, there are many theories about junk DNA, which is really a catch-all term for many different types of classes of DNA sequences that are either non-protein coding or that we don't yet understand. And so actually that's one of the purposes of this paper that I said was actually just came out last week in Science Online, so it's not yet in the print version, where we argue, and thanks for bringing this up because I didn't really have time to tuck this in, that the fact that we've showed that approximately 5% of the genome consists of transposons which contribute all an essential role to DNA rearrangements means that this portion of the genome that has been previously thought of as junk, the 5% at least that's derived from this class of transposons, there's probably others as well, actually play an important role right before they expire. So this particular cell has developed a wonderful symbiosis kind of between the transposons and the host genome, where they're retained because they've become functionally essential. If we knock out the transposons, then the cell can't undergo its proper DNA rearrangement. So this is a remarkable case where the transposons have been not actually domesticated, which means they haven't been modified, but they're just wholesale providing a function. And the fact that you require the use of all of those families of transposons, TBEs 1, 2, and 3 together, supports the idea that the whole fraction of transposase that's available in the genome, all of its many thousands of copies, are producing active transposase that can contribute to the rearrangement process that's essential during development. So usually junk DNA is a catch-all term, not really used by people explicitly in the field because it doesn't encode, it doesn't embody a content. Junk DNA is just what we don't know about. But it suggests that sometimes there's a mutualism that's taken place between the junk DNA and the broader organism in which it lives. It's not just a selfish DNA parasite. And so we think that's why, for instance, just eliminating TBE1 or 2 or 3 had no effect. But the fact that we had to eliminate all 3 to see an effect that impacted genome rearrangement is consistent with the additive need for lots of transposase. And you get that from having lots of transposons. So the human genome is approximately 50% transposon DNA as well, but most of it's not active. And so we don't know if it has any roles other than in having shaped the architecture of the human genome over evolutionary time.
Maybe a lighter question. If it's true that our children are what we eat, then they're also what we do. So if you develop a skill set, it could have a bearing on the genetic makeup of your child. Is that extrapolating too far?
It might be that's getting to a subject that's more how epigenetics interacts with neurobiology, which I have to say I don't know very much about, and I think there just isn't much known about how information can be transferred that way. But if a methylation mark or a chromatin mark on DNA influences learning and memory or any other aspects, then such a mark could possibly be transferred to the next generation if it also infiltrates the germline. So sperm are known to pass RNAs as well.
We generally call environmental evolution, then could be passed on.
Mm-hmm. So you have to be less risk-taking, right?
This is just a question from an expert like in genetics. There's this big study looking at different diseases, cancer, diabetes, Alzheimer's. They were looking at many people with these diseases, trying to look for genetic markers so you could have a test. And what they found is that all of these different family lines of people with these diseases, the genes are, the mutations are completely different. So David Goldstein at Duke says that we're wasting our time. Genes are really not important in causing disease. What's your opinion? How much does genetics matter in causing a disease?
Well, actually, so I just heard David Goldstein speak too. I think one of his points is that if you use this genome-wide association approach to try to find a gene for X, what you tend to do is you find many genes right off the bat. And then there's some phenomena, whatever X is, for which there's also, interactions of many traits or epigenetic ways in which it can be modified. And you won't find all of those. So there's very, very complex phenotypes which you can't apply straightforward genome-wide association studies to. And then there's the ones that are inherited as single gene or 4-gene effects. And so there's one of his points is actually that there's a lot of low-hanging fruit which you get right off the bat. And so he tries to argue that you don't need to go the extra mile to try to get every piece using such genetic studies because you won't find them all. And the reason you might not find them all could be that they might be elusive because they're actually dependent on an epigenetic mark, or the point can be a phenomenon can have mimicry, and so you might have just many different possible genetic systems that produce the same phenotype, not just one, and that often happens too.
But do you think genetics really plays a key role, or how much?
Well, I wouldn't discount that at all. I mean,
I'm just trying to get a feel.
yeah, so as somebody who's recently had 3 offspring, I'm impressed that the first one was genetically tested for 5 standard marker loci and the third for 2,000. So there's an impressive array of single genes associated with severe human disease that are certainly well studied.
I just wanted to comment that Goldstein wasn't saying genes don't matter. What he
Yeah.
was saying is that if you look,
New York Times that said
right, I'm familiar with the article, but what he was saying, because I read his commentary in the New England Journal, is that if you're looking at genes that are involved in complex diseases like, which is a stupid term probably, the diabetes, let's say, or hypertension, there are many, many genes involved. What they were hoping is that there would be common variants that would be responsible for that phenotype in all kinds of people. But it turns out that the common variants have such little effect, each one alone, that they're not very useful. And what is probably the case is that these diseases involve rarer variants, which are harder to find, but have more effect. So there's a bit of a trade-off. You can easily find the common variants that have small effect, but it's hard to find the rare variants that have big effect. But that's not saying genes don't matter.
Well, a lot of these tests.
It's saying there's
are
a point of
getting
diminishing
done.
returns. Yeah.
Tests are, I just gave a talk on Monday about the fact that there's a lot of hype about these gene tests, and they're oversold.
If there are no more questions, let's give a hand to.
Thank you.